Good morning, dear students. So, today I am going to discuss about pteridophytes. So, in the last video, you already studied the classification of pteridophytes, and in this class, one by one, we are going to study examples of pteridophytes in detail, both external and internal character. So, first order is a Psilotails. In that example, Psilotum. So, Psilotum division, Xylophyta, class Psilopsida, order Psilotails, family Psilotaceae and genus Psilotum. Psilotum is one of the primitive pteridophytes, including the order Psilotails. So, it is represented by two species, namely Psilotum nudum and Psilotum felicidum. So, of these two, Psilotum nudum alone is reported from India. So, only in Psilotum only you studied two examples here. In that, only one example you find in India. Psilotum is commonly called as whisk fern. So, Psilotum nudum is an erect plant and it grows in the crevice of rock and soil. So, it's a Psilotum nudum is erect and it grows on a other rock or in the soil, and it is erect plant. So, here you can see the structure of the plant body, Psilotum. So, here it is having a underground rhizome. Here root is absent. So, you find rhizome, aerial branch, the small leaves, and you can see here little bit reddish color, that's nothing but reproductive organ synangium, right? So it's a dichotomously branched, both root and rhizome and stem. So here it occurs in tropical and subtropical regions all over the world. Occasionally it grows as epiphytic on forest trees. So Psilotum nudum is an tropical and subtropical plant, and it grows on a rock or a soil. But Psilotum felicidum is a pendulous epiphytic, grows on a trees. So Psilotum felicidum is an pendulous. You studied in physics, I think. So that's nothing but a hanging type. You find the plant. It is restricted to Mexico, Jamaica, and Florida, Hawaii, and some Pacific islands. Only you find this plant in islands. The plant body of Psilotum is diploid sporophyte. Nothing but it is differentiated into aerial axis and a subterranean rhizome. The rhizome is cylindrical, prostrate, and dichotomously branched structure. So here rhizome is a dichotomously branched, nothing but two at a one point, and it is prostrate, cylindrical. It is hidden below the soil surface or a humus accumulated on tree trunks. So it is hidden below the soil. It is an underground modification, or it may be in the epiphytic. For example, in Psilotum felicidum, it may be hidden in a humus accumulated on a tree trunks. So the rhizome bears small, scaly, and numerous rhizoids here, but no roots. Here roots are absent. So rhizome bears small, scaly, new rhizoids here. And the rhizoids are filamentous and up to 3 cells in height and are covered with a thick cuticle. Here the rhizoids are covered with a thick cuticle and are in a filamentous form. Rhizoids anchor the rhizome in the substratum and draw mineral and water from the substratum. So rhizoids anchor, nothing but attached to the substratum, hold the substratum and absorb the minerals and water from the substratum for their further growth. Some branches of rhizome turn upward and grow into aerial branches. In the rhizome, some branches turn upward and grow into aerial branches. The aerial axis is erect in a Psilotum nudum, and it grows up to height 20 to 25 centimeter, because it is present in it is a tropical and subtropical region. That's why it grows up to 20 to 25 centimeter height. But in Psilotum fellicidum, the aerial axis is pendulous. It is in a high form and grows up to 100 centimeter in a height. Aerial axes are green and dichotomously branched, nothing but your stem. Aerial axis, it is a green color and dichotomously branched here. Its basal portion is slightly rigid and the distal portion is triangular in cross section. While in the aerial axis, the below portion, basal portion is slightly rigid, but while going to the upper, it is in a triangular. When you take a cross section, so tip of the branches has a single tetrahedral apical cell to proceed the growth. So in the tip, it is having a single tetrahedral apical cell for their growth. Photosynthesis mainly concern this aerial axis. That's why the aerial axis are green color, because here leaves are not properly developed. That's why stem only act as a photosynthesis process. The axis is branched. It bears many small scale-like appendages called as leaves here. So there are a small minute leaves are present here. The leaves are arranged in a spiral manner, but they have no vascular supply and stomata. So here in the leaves you won't find a vascular system and a stomata here. That's why stem only act as a photosynthetic process. So, however, small leaf traces are seen in Psilotum fellicidum. Here in Psilotum fellicidum, the small leaf traces are present. Right. In the upper region of the axis, the leaves are replaced by fertile appendages called sporophyll. So, in the last diagram, you saw that nothing but a small reddish color reproductive organ sporophyll, that's replaced by the leaves. Each sporophyll is bifid structure that bears a trilocular sporangium in its axis. In the axis region of sporophyll, it bears a bifid structure and a trilocule. It contains three-chambered sporangium. Inside sporangium, you find a spores. While studying detail, you are going to study. So next comes internal structure one by one. So first we are going to study internal structure of rhizome. The cross section of rhizome shows an epidermis, cortex and a endodermis and a central stellar region. Right, you can see here in a diagram, it is having a outer epidermal region. It is a single layered, it is made up of thin walled cell, and the epidermis is lined with a layer of cuticle secreted by the epidermal cell. So the single layered outer cuticle is present, right? So it contains a chemical substance secreted by cutin and a wax. So rhizoids access from the epidermal cell. So by the epidermal cell, the rhizoids arise. So next comes cortical region. Here cortical region are three layered: outer, middle and inner cortical region. So inner to the epidermis is a cortex. The cortex is differentiated into three zones, nothing but outer cortex, middle and inner cortex. The outer cortex is otherwise known as a mycorrhizal zone. It is made up of thin walled cells. So here outer cortical, nothing but it is thin walled cells, and it is known as mycorrhizal zone because most of these cells are infected with the fungal hyphae, but sometimes infected with the cells may digest with fungal mycelium. So here the outer layer is infected with the mycorrhizal fungal hyphae, that's why they are called as a mycorrhizal zone. But sometimes the infected cell may digest the fungal mycelium. However, in the cross section, many cells contain intracellular mycelium. The middle cortex consists in walled parenchymatous cells. Starch grain are stored in these cells. So in the middle cortex you find a loosely arranged thin walled parenchyma cells. In that starch grains are stored. The inner cortex is dark brown. The last layer of the cortex is the inner cortex. It is dark brown in color due to the storage of phlebophins. Phlebophins, nothing but it is a phenolic substance and it is a dark brown or a reddish color. The cells are however thin-walled and parenchymatous. So inner cells also parenchymatous and thin-walled, but here you find a phenolic substance is present. That's why it looks dark brown in color. So next comes endodermis, followed by cortical region is followed by endodermis, single layered, and it is made up of elongated parenchymatous cells. The endodermal cells are thin walled and have distinct Casparian strips in the lateral walls. So in the lateral walls of the endodermis, we find a Casparian strips there. Next important is a stellar region. So here the stele is a protostele and it lies inner to the endodermis. The outer boundary of the stele is pericycle. It consists of thin-walled cell. So the here stele is a protostele, normal, nothing but a central you find a xylem and surrounded by a phloem here. And the outer boundary of the stele you find a here thin-walled pericycle. The inner to the pericycle there is a vascular tissue, nothing but xylem and phloem. Central core of xylem and surrounded by a phloem here. The xylem consists of tracheids having scleriform thickening and it cannot easily be distinguished into protoxylem and metaxylem. Here protoxylem and metaxylem are not distinguished because here tracheids contain a scleriform thickening here and phloem completely surrounds the xylem. It consists of elongated cells. So phloem consists of elongated cells and here it is surrounded by xylem. Here pith is absent. This stele is known as a protostele. Protostele nothing but a xylem is surrounded by a phloem. So it's an internal structure of a rhizoid. It contains a outer single layered epidermis and it is covered with cuticle, and inner to the epidermal cell you find cortical region, that is 3 layered, outer one mycorrhizal zone and a middle cortex parenchymatous cells and a inner cortex which contain a phenolic substance dark brown color, and it is followed by single layered endodermal cells. Inner to the endodermal region is a vascular tissue. It contains outer thin walled pericycle and followed by a protostele. The xylem is central and it is surrounded by a phloem. So this is a brief study of a rhizome. So in the next video we are going to study the stem, internal structure of a stem. So I think you all understood this. Thank you.